Sorry. Cool. Okay, let's see. All right, guys. Sorry about that. I am a grandma when it comes to technology. Oh. All right, how are we feeling about immuno? Good, bad, indifferent? Okay, well, so we are going to get started. We're going to try to cover high yield stuff as well as confusing stuff. If we don't get through all of it, we sent you guys our PowerPoint, but we're going to try really hard. So first is, so we're going to go in the lectures that you had this week, like in order of them. So first is the really confusing lecture by Dr. Swanson. Unless you guys thought it was easy, that's great. But I remember last year, it was straight chaos for our class because everybody watched it. And then afterwards, they just did not know what to do. So before we start, we're going to do a little pre-check. So does anyone know where B cell development occurs? Just feel free to shout it out. Bone marrow, okay. And then what about selection? Like positive and negative selection? Nope, it's the bone marrow. Yes, so this is different than T cells, which do it in the thymus. And then what are the 3 APCs? Perfect. And then lastly, what marker do all B cells have? This is kind of a tough one. I heard it over there, CD19. This will help you in heme, so make sure you guys remember CD19/CD10, CD20, because it'll help you with all the lymphomas and stuff. So this was one of the most confusing slides that I whatever. Okay, anyways, I'm just gonna start. So I'm just gonna point some like important things out. So first point, CD10 expressed in immature early developmental B cells. So developmental B cells are ones that don't have a B cell receptor yet. But as they mature into a mature B cell, they start to express CD20 and they start to lose their CD10. So that's important to know. And also knowing when RAG is expressed. So did you guys have VDJ rearrangement last week? Yeah. So that's a rearrangement enzyme. So it's going to happen whenever you're having rearrangements, obviously. So that's going to be in the pro B cell, where it's going to undergo heavy chain rearrangement. And then it's going to stop. And it's going to stop to check to make sure that heavy chain is correct and that it's functional, because there's no point in putting a light chain on top of a heavy chain that's not going to function anyways. So we're going to go into the mechanism of that in the next slide. But so while it's in this stage and it underwent heavy chain rearrangement, it's going through a check. It's going to undergo proliferation. And since it's going through proliferation, there's going to be no RAG expression. Does that make sense? Okay. And then once it passes this pre-check, it's going to go and rearrange light chain, and then RAG is going to be expressed again. So those are the main points. I think that, and then knowing that an immature B cell expresses IgM, but a mature B cell expresses both IgM and IgD. So if I confused you, I gave you some takeaway slide. But this is kind of an important slide just because it matters clinically to one of the disorders that you guys learned about. So this is the transition from a pre-B cell receptor to a B cell receptor. And the way that it works, like I said, is it's going to check to make sure that that heavy chain is functional before it rearranges its light chain. So the way that it works is it takes the heavy chain that it rearranged and it attaches it to a surrogate light chain, SLC, and it has 2 parts, which is the VPREB, which is the variable region of the light chain, lambda 5 constant region. I don't really know if that's important, but what it does is it takes the surrogate light chain, attaches it to the heavy chain, puts it up on the cell surface to see if it's functional. If it passes that check, then the B cell receptor will be internalized again and will undergo light chain rearrangement. If it doesn't pass that, it will get internalized. It will retry the VDJ arrangement and then try again. Does that make sense? So the clinical relevance of that is the Bruton's A gamma globulinemia, because there's this enzyme called BTK, which please do not memorize this pathway. There is absolutely no point for you to do that. Just know that BTK is important in the transition from a pre-BCR to a BCR. So it's important in ending heavy chain rearrangement and beginning light chain rearrangement in the BCR. So if you don't have that, then you can't fully make mature B cell receptors, which is why in this disease, you don't have transition from pre-B cell to B cell, so you don't have any immunoglobulins. That's why it's A-gamma globulinemia. Does that make sense? Any questions so far? Beautiful.
How about online?
Oh, online, do you have any questions?
Looks like our chat's empty.
Okay. No, that's okay. All right, so this is pretty, a little bit easier than the last thing. But so then what happens is once you rearrange and get a B cell receptor, it's going to undergo positive and negative selection. And it's really important to know what positive is and what negative is. So positive selection means that you want the B cell to do something. You want it to be able to bind to an antigen, and you want it to bind tightly. So the B cells that bind tightly to the antigen or their receptor, those pass positive selection, and they get a survival signal from the antigen presenting cell that it binds to. If it can't bind to the antigen presenting cell, then it will undergo apoptosis. And then once it passes positive selection, it'll go to negative selection. And the way that I think about it is negative selection means it's something that you don't want the B cell to do. So negative selection, you don't want the B cell to do it. You don't want it to bind to self-antigens. Therefore, if it binds to self-antigens, undergoes apoptosis. If it passes this, it becomes a naive B cell and it leaves into the blood, but it still hasn't seen its antigen yet. It's only seen an antigen presenting cell. So it hasn't been exposed to any pathogens yet. That's why it's called naive. Once it gets exposed to a pathogen, then it'll be called a memory B cell or an effector B cell. So that's just the terminology there. Okay, and then B cell activation, it can be T cell independent and T cell dependent. I would say that this is important to know. So knowing that T cell independent pathway is just polysaccharides, so bacterial polysaccharides. And the way that it works is the polysaccharides are polymeric and they're long enough to where they can cross-link with multiple receptors on the B cell, which allows it to overcome the signal that's needed for the secondary signal for T cells. And the other thing that's really important to remember is that since it's not binding to a T cell and it's T cell independent, there's not going to be any kind of like somatic hypermutation or class switching. So it's always going to be IgM, and the affinity is going to be pretty low because it's not trying to change the affinity of anything. It's just binding to whatever it can. However, in T cell dependent, I think it's important to know the two receptors that you need. I think that's very high yield. So knowing that, first of all, you need two things to happen. You need the MHC protein on the B cell to bind to the T cell receptor with its antigen. And then the second thing you need is an interaction between CD40, which is on the B cell, and CD40 ligand, which is on the T cell. If you don't have these interactions, then you will get another disease that you guys know about that I'll talk about in a second called IgM hyperimmunoglobulinemia, I think. So make sure that you guys know that you need both of those interactions. Okay, I'm not going to go over this, but I will say that I do remember him asking questions on the functions of each of these cells. So the little red things down here, I want to say it was maybe this one, but I'm not 100% sure. So I would just say give that a look over if you want to point. If not, just throw it out. But on to the next. Okay, so we're going to go over somatic hypermutation and clonal selection. So once the B cell gets activated by a T cell, this only happens in T cell dependent activation of B cells. It will undergo somatic hypermutation and clonal selection. And this is a way for the B cells to take their non-specific antibodies, or not non-specific, non-specific receptors, and change them around to become more specific and selective for the antigen that they want. I put in this little saying at the bottom. It's from USMLE. If you guys use that, it's very helpful. If not, it's not. But I thought it was like an easy way to kind of break down what somatic hypermutation is. But yes, and so the other thing that's really important is that somatic hypermutation needs the activation-induced cytidine deaminase, or the AID enzyme. That's important for both somatic hypermutation and class switching. And then once it undergoes somatic hypermutation, it tests its receptors to the antigens. Once it finds one that binds really tightly, it undergoes clonal selection. So the ones that bind really, really tight to the pathogens or the antigens, those are the ones that are going to keep proliferating, and they're going to proliferate at such a high rate that those are going to be the antibodies that you make. Does that make sense? Wonderful. So you need to know this mutation. He gave you a slide where there's like all of the different kinds. I don't know if you need to know that. I'm not going to say that you don't because I don't remember, but I do know that this comes up a lot. So I would definitely know that. And so the reason why hyper IgM syndrome happens is because you don't have the ability to class switch or undergo somatic hypermutation. And like I said earlier, you need either of these, well, you need both of them to undergo B cell activation by T cells. So if you don't have those, then you can't undergo class switch or somatic hypermutation because the signal from the T cell is what signals the B cell to undergo class switch. Does that make sense? Okay. And if I'm going too slow, you guys can just tell me to keep going faster. All right. I would say that you should understand this. You'll probably get 1 or 2 questions about it in your time in immunology if you haven't already. So just knowing that your primary antibody response is slow. It takes a week to 2 weeks after exposure to the infection for you to even start an immune response with antibodies. But the good thing about the primary response is that at the end of it, you make these things called memory B cells, and those are the ones that help you in your secondary exposure. So your secondary exposure is fast, and it's also more robust because you have those memory B cells, and then you also have the naive B cells that are also coming to this site of infection or whatever it is at the time. So just understand that. And then we're going to go T cell development, which I thought this was really funny because I don't know if you remember this, but literally like 99% of the T cells that are in the thymus die. So it's like not ineffective, but it's fine. Okay, anyways, so we're going to do another pre-check. So where does T cell development occur? Like where are they made? Bone marrow. But where do they undergo selection? The thymus. Nice. And then which interleukin is important for the development and survival of T cells? Yep, I heard it over there. IL-2. And this is a really important one to know because it's needed for all T cells, but it also is especially needed for the activation of CD8 T cells, because if you don't have IL-2, you cannot activate the cytotoxic T cells. And then lastly, which one suppresses T cell activity? Beautiful, you guys are ready. IL-10, I would know that too. All right, so we're gonna go through this. It's pretty self-explanatory. Maybe, I don't really know if it is, but I'm confused on it. So once you have a T cell, an immature, naive, pre-T cell travels through the blood from the bone marrow to the thymus. The first place that it goes is the subcapsular region. And here is where you have triple negative cells. And so these triple negative cells have high BCL2, low FAST. That means that they're anti-apoptotic, they're pro-life. So that sounds really controversial. So they're just going to proliferate. I didn't mean it that way, sorry. They're going to proliferate a lot, which is why you have so many T cells in the thymus. And so once they do this, they're going to proliferate. Then they're going to start expressing BCL2. They're going to downregulate BCL2, upregulate FAST, which means that they're pro-apoptotic. So a lot of them are going to die. So they're going to move to the cortex. And here's where they become double positive cells, although they are actually triple positive cells. But CD3 is the marker for all T cells. So CD8 and CD4 T cells will both have CD3, like marker for CD3. So here is where they undergo positive selection. So positive selection, cortex. This is where you want the T cell to do something. You want it to be able to bind to an MHC receptor. So if it does this, it's called self-restriction. Drescher said that. I don't know if that's, I don't know. What if she puts it on an exam? I don't know. But anyways, if it does that, then it passes positive selection and we'll move on. If it doesn't pass that, it's gone. It undergoes apoptosis. So the way that T cell becomes CD8 positive or CD4 positive is dependent on the MHC receptor that it binds during positive selection. So if it binds an MH1, it's going to become a CD8. If it binds MHC2, then it's going to be a CD4 cell. So the ones that pass positive selection go into the medulla, and these ones are going to undergo negative selection. So negative selection, medulla. Single positive cells, they're either CD4 or CD8 cells. They've decided their fate. And so negative selection, like I said, you don't want it to do something. You don't want it to bind to self-antigen. If it binds to self-antigen, then it will not pass negative selection and it will die. But if it doesn't bind to your self-antigen, then it becomes a T cell. And so the way that it works, I would make sure you guys know that about that AIRE thing, the AIRE, where it's your body that's presenting the antigens, not actual pathogens. So it's the epithelial cells in the thymus that are training your T cells and making sure that they aren't binding self-antigens. It's not actually real antigens. So this is important to know because once it passes positive and negative selection, it gets released into the blood as a naive T cell, which means the same thing as a naive B cell. It means that it hasn't yet seen its pathogen, antigen thing, whatever you want to call it. So neither of them have seen their pathogens yet. But once they do, they'll become whatever they want to become. But the other thing that's really important to know is that the cytokines are what determine what kind of T cell it will become later. So if it's CD4, there's a lot of different subtypes of CD4. So if you expose a CD4 T cell to like IL-12, for example, that CD4 T cell will become Th1. If you expose it to IL-4, then it will become Th2. So whenever a T cell initially leaves the thymus, it's Th0. So it has the opportunity to become Th1, Th2, or Th17. Tregs, on the other hand, leave the thymus as Tregs. They are already differentiated. They're positive for CD4, CD25, and FOXP3, which is a transcription factor. But I think the most important part to know is that they secrete IL-10, which we know that inhibits the immune system via T cells, and they leave the thymus as Tregs. So know that they don't become Tregs later, they become Tregs in the thymus. All right, and then the other one is Th17 cells. Yes. No. So then Th7, actually I'm not sure. I'll get back to you on that. I'm not gonna say no and just, okay. Anyways, Th17 cells. So the cool thing is that there's an interplay between these Th17 cells, these Th1 cells, and these Th2 cells. And so Dan, I think you're gonna go through this later.
Yeah, I'll go through this in more detail.
Yeah, so I'm just gonna scratch the surface. So know that TGF beta and IL-6 are needed for the formation of Th17, but you also have to have IL-23 in order for them to survive. So you could make them, but then they'll die if they don't have IL-23. And then these are the things that they secrete. They're secreted by interferon gamma, which is Th1, and IL-4, which is Th2. So both Th1 and Th2 will inhibit Th17 cells. Does that make sense? It's kind of like the arrow thing that Drescher had between the interplay between the Th1 and Th2 response. Does that make sense to everybody? Okay. Yes.
Something I'll say real quick. So it gets really complicated when you start looking between the different regulations of the T cells and how they regulate each other. I would say that's less important than knowing what each type of T cell secretes and what cytokines activate that T cell. I would focus less on how they kind of affect each other. You know, if you know the subtypes, I think that's enough. So
I agree. All right, this is a cool table. It's from USMLE if you guys want it. I'm a table learner, so I included it. So now we're gonna talk about MHC. So first, we're just gonna go through the primary organization of it. So MHCs are inherited from your parents. You get 3 MHC class 1 and 3 MHC class 2 genes from each parent, and they're all located on chromosome 6. And so they don't really undergo rearrangement. Some of the times they do like crossover or whatever, but they're co-dominant. So you don't have like one that's dominant over the other. You're gonna express all of the ones that your parents give you. And then they're polymorphic. They like to ask you about that. They're very polymorphic, which means that there's a lot of different variations within a population. So it gives certain people advantages over others in certain types of like infections and whatnot. And know that MHCs are required in order to activate T cells. So class 1 is HLA A, B, and C. And I'm going to tell you all the high yield stuff here, but know that only the alpha chain is encoded on chromosome 6. Know that there is no beta chain that is encoded for class 1 on chromosome 6. It's on chromosome 15, and the beta chain is called beta 2 microglobulin. So what this means is that the variability of MHC only comes from the alpha chain, because all MHC class 1s have the same exact beta chain, beta 2 microglobulin. Does that make sense? Cool. So then class 2, on the other hand, has these ones. I'm not going to read them to you. But the cool thing about this is that they have both alpha and beta chains that are encoded on chromosome 6. So this means that class 2 has way more variability because they get different combinations between alpha chains and beta chains because they're both unique. So knowing that class 1, only alpha, class 2, alpha and beta.
Yep. And if you look at the picture too, this is a good image from first aid, but you can see the alpha chain is what's in contact with the T cell receptor, where this is beta and alpha. So this would be MHC class 2.
Yes, and make sure you guys know this in words and also can look at a picture and recognize which one they are, because they love to give you pictures of T cell receptors and of these and ask you which ones they are. So there's this cool thing called linkage disequilibrium. I'm sure it's pretty self-explanatory. I just left it in here because I figured it was kind of high yield, just that certain genes are inherited together more than others. And that means that certain diseases can be associated with certain combinations of MHC. But this is important. These are the 3 things that you need in order to activate a T cell. So you need an antigen peptide that's broken down into a peptide, and it has to bind to a self MHC. It can't bind to someone else's MHC. And then it has to interact correctly with the T cell receptor. So I do remember getting a question on this, I think. All right, so we're gonna go through the structure really quick. This is all really high yield. They'll ask you a lot of questions about this. So class 1 MHC, like I said, it's 3 alpha chains. Beta chain is beta 2 microglobulin. Beta 2 microglobulin is not inserted into this plasma membrane. So only the alpha chain is inserted, which means that class 1 is way more unstable. That's why class 1 is so unstable, is because the beta 2 microglobulin is not inserted into the membrane. The other thing to know is just that class 1 binds smaller peptides instead of larger peptides. So I think that's the major thing. And knowing that the peptide binding cleft, like Dan said, is between 2 alpha chains.
Have you guys ever been to or know about Cape Cod? When I look at MHC class 1, I think of Cape Cod. It's like an elbow. It sticks out from Massachusetts. So that's kind of right, like kind of there. You can kind of see it. So I don't know. My grandparents live there, so sorry. But that's how I think about it.
They're never going to forget that.
Never.
Beautiful. All right, MHC class 2. It has 2 alpha chains, 2 beta chains. Alpha and beta are both inserted into the membrane, which means class 2 is way more stable than class 1. And the peptide binding cleft is between alpha and beta chains, and then they can accommodate for larger peptides. So knowing which one's smaller peptides, which one's larger peptides is important. And then knowing how they bind to the peptides is also important. So knowing about the anchor residues and the peptide motifs. So the anchor residues are what allow MHC molecules to bind to a bunch of different like pathogens, because they only need certain amino acids to be in their binding cleft. And then everything else doesn't matter. As long as they have those anchor residues, then it can bind to anything. But the important thing to know is that it can only bind one thing at a time. So you can only have one peptide in the binding cleft at a time, but it can bind a whole different array of peptides overall. And then also, Drescher stressed this like 7 times, maybe 8, knowing that MHC, most of the peptides are self-peptides, not pathogens. So make sure you guys just commit that to memory real quick. All right, antigen processing and presentation. Is everyone okay so far? Do we have any questions? Am I going too slow? Too fast? Good, cool. All right, online people.
No chat, we're good.
It's killing it. All right, so antigen processing and presentation. So the antigen can go through one of 2 pathways, MHC1, MHC2. That's what we just talked about. MHC1, intracellular, viruses, stuff like that. MHC2, exogenous, mostly bacteria, whatnot. But knowing that what determines which pathway a pathogen goes through is based on its location and nothing about the actual pathogen itself. So if it's like extracellular, it's always going to go through MHC2. If it's always intracellular, then it's going to undergo MHC1. Does that make sense? That's important to know.
Good example of this is bacteria. Some bacteria, most of them will live extracellularly, but tuberculosis is an example that lives inside cells. And so that would actually be presented on MHC class 1 because it comes from inside the cell, but it's a bacteria. So that's just an example. So the location is more important than the type of pathogen.
Exactly. And then, cool. So we're going to go through this really quick. I might sound like it's detailed, but I have everything typed for you on the next slide. This is class 1. So the pathogen is going to be extracellular or intracellular? Intracellular. Nice. So since the pathogen's already intracellular, you don't have to bring it into the cell. So the first thing that happens is, say that it's a virus, it's going to undergo replication. It's going to synthesize proteins, and these proteins are going to be recognized by the cell as bad. They're going to get ubiquitinated and sent to the proteasome, which is like LMP proteasome, I think. But it gets processed through these proteasomes, and it cleaves that protein up into smaller peptides. But some of the peptides that are made from the proteasome are too large. So cytosolic peptidases will make them smaller. That's very important to know because I remember that on a quiz. Also, I can't guarantee that any of this will be on there, but just prior memory. Anyways, so peptidases cleave them smaller because MHC class 1 can accommodate for small peptides, not large ones. So you need them pretty small. So the peptidases make sure that they're small. So once you have peptidases that are the right size, they're going to go into the rough endoplasmic reticulum via TAP. So knowing TAP is also important. And once it enters the rough endoplasmic reticulum, you have this cool protein called tapesin that pretty much just anchors it. So it doesn't go anywhere. It can't run away. But at the same time that all of this stuff is happening with the pathogen, your rough endoplasmic reticulum is also synthesizing MHC class 1 proteins. So you know that MHC class 1 protein is made up of an alpha and a beta. The alpha is that 3 alpha chains. The beta is the beta 1 or beta 2 microglobulin. So they're synthesized separately. And without beta 2 microglobulin, the alpha chain is very unstable. So it's bound to a chaperone protein called calnexin, which I don't really know if you need to know that. So it's bound there. Once beta 2 microglobulin joins with its alpha chain, it'll get transported to the tapesin complex, and it will be like the peptide / antigen will be inserted on the MHC class 1. And then once they're like bound together, they will become exocytosed, and then they'll be presented on the cell. So here I bolded and underlined things that I think are important. For some reason, I just think calnexin is important, but I don't know why. Also knowing that you need TAP1 and TAP2 in order for it to function. So if you have a mutation in either of these 2 proteins, then you can't make MHC class 1 proteins. Do you have something to say?
Yeah, real quick, I think Cassie mentioned this already a little bit, but most of the proteins presented on MHC class 1 are not pathogens. They're from just your normal cells. So, and that's really important when you think about like tumor cells who, or even, in viruses, but tumor cells will present sometimes abnormal antigens, and they'll also sometimes downregulate MHC1, which is where NK cells come in. But it's important to know that most of the stuff on MHC1 is host. So
yes, perfect. All right, MHC class 2 pathway. I also have it typed out for you. But I'm just going to tell you the high yield stuff. So first of all, the pathogen's extracellular. So you have to either phagocytose it or you need to do receptor cell mediated endocytosis. So you have this vesicle, it fuses with the lysosome that has some proteins in it that I typed out on the next slide that you probably don't need to know. But the acidification of the lysosome is what breaks the pathogen down into small peptides. And so while all of that is occurring in the rough endoplasmic reticulum, you're also synthesizing MHC class 2 proteins. So you have the alpha chain and you have the beta chain, and they are, once they're both synthesized, they're combined together, and you have this thing called an invariant chain, which is very important to know, that it binds to the MHC class 2 protein, and then it gets cleaved into a clip fragment. So knowing clip and invariant chain are important. And then knowing that the clip fragment is important for preventing any other peptides from binding to the MHC class 2 on its way to this cool vesicle over here. So it just blocks the binding site. And then once the two vesicles will fuse, you have HLA-DM, which is the non-classical type or class 2, which is also important. I do believe that was a question. That HLA-DM is what removes the clip protein and allows MHC class 2 to bind to its antigen. And then it gets exocytosed and presented. Does anyone have any questions on that? Yes. Yes. I believe so. I don't think so, based on what she said. No. It gets clipped on the way to the lysosome. Sorry, I'm a little rusty. Yeah, we have a question too. Can you repeat question into mic? Yes. She asked if, or he asked, sorry, if the invariant chain gets cleaved in the rough endoplasmic reticulum or if it gets cleaved on its way to the endolysosome. And the answer is on its way to the endolysosome. Okay. Cool. So yeah, here's everything that you need to know. I underlined and bolded the things that I think are important. And then lastly, we're going to do cell-cell interactions before Dan takes over, which will be more fun. Okay, so we're going to talk about this really quick. So activation of a naive T cell. Naive T cell means it hasn't yet seen its pathogen. It means that it's a baby pretty much. And so this is really, really, really important to know that a naive T cell can only be activated by a professional antigen presenting cell. And a professional antigen presenting cell is one that has high amounts of MHC and B7. So dendritic cells are the prime example of it. They're pretty much the main professional ones. But the sequence of events in the activation of a naive T cell matter. They're very important because if they're not in order and if they're not in the right time, then the T cell will not undergo activation. So the first thing that happens is the antigen, obviously processed, presented on MHC. It binds to the T cell receptor, but as it binds to the T cell receptor, it must receive immediately co-stimulation between B7, which is on the antigen presenting cell, and CD28, which is on the naive T cell. If this doesn't happen, if this step is missing, then the T cell will undergo anergy and it won't undergo activation. So you need that. That's the most important part of this. And then after that, you get the CD40L with the CD40 on the antigen presenting cell. Does that make sense? Cool. All right, for a memory T cell, this means that it's already seen its pathogen once before, if not more. So the sequence of events don't necessarily matter as much. So it doesn't need that immediate stimulation by the B7 CD28. It will bind with the MHC, and then you'll get the CD40L ligand attaching with the CD40 on the antigen presenting cell. And then this will induce B7 expression if the cell isn't, if the antigen presenting cell isn't already professional and expressing it. And then you'll have that interaction, and then you'll have stimulation of the T cell. All right. I'm sure most of you guys already know this because you guys went over it with NK cells. This is pretty much the same exact mechanism. It's for cytotoxic T cells. The most important things to know are right here, perforin and granzyme B. They're red, they're underlined, and they're bolded, which means that that is very, very, very high yield. So just know that perforin punches a hole in the target cell. Granzyme is the one that enters the cell and makes it undergo apoptosis. I'm just not going to spend much time on that. Next one, this is also very, very high yield to know. So the FAS-L pathway. So FAS ligand is expressed by the cytotoxic T cell, and then all other cells will express the FAS receptor. So the interaction between these two things will trigger apoptosis. But the most important thing to take away from this slide is that it's the cell expressing FAS, not the cell expressing FAS ligand that dies. So make sure you guys know that.
A lot of tumors will downregulate FAS to avoid killing by CD8 T cells.
Yes. So knowing that's really important. All right, I'm not going to spend much time on this. I'm just going to tell you the things that I think are important. Just knowing that there's two separate types of memory T cells. There's the central ones, which are important for the second wave of a response, and the effector ones, which are important for the primary, really quick response that you get. And then knowing the requirements for survival may help you on a quiz or exam. And then this is a pretty important topic, but it's pretty self-explanatory. So whenever you're first exposed to an, like some kind of infection, say that you have these 4 epitopes, that I took this slide straight from Drescher. So you have these 4 epitopes, say that you get a second infection, you'd still take the epitopes that were from your first infection, and those are the ones that you recognize. And then if you get infected a third time, you're still going back to the first primary infection. Your secondary response or your secondary exposure does not change the epitopes that your body will prefer to like react to, if that makes sense. Cool. All right, and this is my last slide. So both of these, activation-induced cell death and cytokine withdrawal, are both forms of apoptosis, but they have different cells that are causing the cell to undergo apoptosis. And the activation-induced cell death, it's FAS and TNFR. And cytokine withdrawal, it's BAX and BAC, and it's because of the lack of release of IL-2, which we know is important for the survival of T cells. And then T cell exhaustion just means that they're tired. So, all right, you got it, bro.
Cool. Okay, how are we doing? Do we need jumping jacks or something?
Sorry, that was a lot of talking.
Oh, it's okay. I wasn't like trying to say you're boring or something. Okay, so I have another meme here that you can look at. So that's what goes on in your cells. Okay, so how are we doing online? No comments? Okay, good. Okay, this, you guys know this, I hope. So your innate immune. So anyway, my goal is going to be to try to synthesize some of this stuff for you and consolidate it, because I think it's easy to think about immunology in different compartments, but when you try to bring it all together, it can be really confusing. So I'm going to try to, a lot of this stuff actually comes directly from Dr. Drescher's lecture on the immune response. It's actually, I thought, a pretty helpful one for me to kind of synthesize some of this. So I'm going to go through a lot of that with you, and hopefully you leave less confused than when we start, but no guarantees. To click. Okay, questions. So first, we're going to talk about how the innate immune system will respond to a viral infection. This is going to be a review from last week, probably, okay? So what innate immune system receptors do viral PAMPs, what do they bind to? What's the name of that receptor class? Anybody know? TLRs, good, toll-like receptors, very good. Okay, there's 4 TLRs that are important in viral response. What 4 are they?
High yield.
3, 7, 8, and 9. Good. 3 out of 4 over there. Pretty good. Yeah. So those numbers they like to ask you. So know those 4 for viral. Okay. So where are these TLRs located inside cells? Endosomes. Very good. Yep. It makes sense, right? Because viruses get in the cell. We don't need them to be on the cell membrane. Okay. So which cytokine is produced by the macrophages and dendritic cells in response to TLR 3, 7, 8, and 9 activation? It's a class. Type 1 interferons, anybody? Type 1 interferons. Good. I heard somebody said 1. That's good. 1, yeah, that's not bad. Okay, so type 1 interferons, and that encompasses like alpha and beta interferon. There's interferon gamma also, which we'll talk about. That's different. So there's this antiviral state thing. This definitely came up on a quiz. So there's 3 things that type 1 interferons do that induce this antiviral state. Does anybody know? That's 1. I didn't list that, so there's actually 4. Sorry. Upregulate MHC, excellent. And there's 2 cytokines that are produced by other cells. Okay, so the first one is IL-12. Okay, we get IL-12 production by dendritic cells and macrophages. Okay, and then the other one is we get interferon gamma production by NK cells. Okay, so these type 1 interferons will bind to dendritic cells, macrophages, and NK cells and cause them to produce IL-12 and then interferon gamma. Okay, and this is important because what do IL-12 and interferon gamma do? They induce TH cell differentiation to TH1 subtype. Okay, and TH1 subtype is intracellular pathogens, right? So viruses and intracellular bacteria for the most part. There's exceptions, but as a general rule, that is kind of how the innate immune system activates the adaptive. So hopefully that is helpful. Okay, so this is just looking at those intracellular TLRs and those endosomes. So 3, 7, 8, and 9, they all respond to different components of viral, either DNA or protein. But you don't have to know that. Just know the numbers is fine. Something else is that this should be in intracellular. When we're looking at this pathway here, this is called the TRIF dependent or MyD88 independent pathway. This is what's responsible for type 1 interferon production and inducing that antiviral state. This dependent pathway we'll talk about when we talk about bacteria. That's more activated with bacteria. Viruses can activate this pathway, but it's to a lesser extent than this one. Okay. Okay, this is from first aid. Really like first aid. It's my friend, my best friend sometimes. Looking at kind of this whole process, I like pictures. I don't know about you guys, but it helps me. So remember, our type 1 interferons are up here, down here. Okay, what are they doing? They're going to the macrophages or dendritic cells, causing them to produce IL-12, okay? Or they're going to the NK cells, causing interferon gamma production. What do those do? Those induce differentiation of naive CD4 T cells to Th1 subtype, okay? And then Th1 subtype will produce interferon gamma and IL-2. What does interferon gamma do? It does macrophage respiratory burst increase, which helps macrophages kill intracellular pathogens. Really good for bacteria. IL-2, on the other hand, we talked about promotes CD8 T cell activation and survival, which is really good against viruses. Okay. So this is a good picture to stare at if you like staring later on. So, okay, how are we doing online? How are we doing here? Good, love it. Okay, now we're gonna talk about bacteria. Okay, what happens with the innate immune system here? So bacterial PAMPs will bind TLR 1, 2, 4, 5, 6. So all the TLRs that aren't viral. Okay, you don't need to know the numbers for this. They like the viral one better. But what is the signaling pathway that's activated? What's the name of it? MyD88, very good. Dependent, okay? It's dependent on MyD88. Good. Okay, so next, this stimulates production of pro-inflammatory cytokines. There's 4 big ones, okay? This is a bit of a stretch, this is a tough question, but can anyone think of a cytokine that's produced? IL-1, good. IL-6, yep. TNF alpha, good. IL-8, did somebody say? That's very good. And then what's like the one that acts on the bone marrow? That is a longer name. Yep, very good. GCSF, GM-CSF. Nice, you guys are crushing. I didn't put IL-6 on there because I forgot, but yeah, those 4 are big though. IL-1, TNF alpha, IL-8, and GCSF, GM-CSF. So these are produced by those dendritic cells and macrophages that had those bacterial TLRs. So 1, 2, 4, 5, 6 activated, okay? So what do those do? Let's figure it out. So what does GCSF, GM-CSF, what do those do? Very good. Go to the bone marrow and induce neutrophil differentiation from phagocyte precursors. Okay. GCSF, GMs, there are different steps in the pathway, but I think it's sufficient to know that they're involved in neutrophil differentiation. Okay. What do IL-1 and TNF alpha do in regards to endothelial cells? They do a couple of things. Good. Permeability. And then what do they upregulate? Good. Yep. Selectins. So the ones I found were E-selectin and ICAM1, but it probably has a role in all of them. So there's P-selectin and there's VCAM1 as well. But those are the ones I found online. So, but yeah, so what do those do? So those grab neutrophils and they grab lymphocytes later on out of the blood and help them get to the site of infection, right? So, and lastly, I spoiled it. What's IL-8 do? Neutrophils. Good. Yeah. So IL-8 neutrophils, that's really high yield. Neutrophil chemotactic. So neutrophils will follow the IL-8 gradient. The more concentrated it becomes, they will follow that. Okay, I already said this, but viral infections also induce this MyD88 dependent pathway, but to a lesser extent than bacterial. Okay, but it still does happen. Okay, so this is like leukocyte adhesion. So this is just kind of summarizing everything. So you have your macrophages here secreting IL-8, secreting those pro-inflammatory cytokines here that increase vascular permeability and upregulate selectins, ICAM, VCAM1. The IL-8 will induce neutrophil. It's a chemotactic, so neutrophils will follow the IL-8 gradient, and then they get into from the blood vessel into the tissue where they need to be to fight the infection. Okay.
Also, just a side note, do you guys remember that graph where it has like E-selectin and ICAM? Know which ones peak and when they peak in the order of them peaking. That's really important.
I'll buy you all ice cream if it wasn't on your quiz last Friday. So.
Oh, sorry. It'll be on the final too.
Was it on your last quiz? I don't have to buy you ice cream then. We love that. Okay, adaptive immune response. So we talked about innate immune response. Okay, let's transition to the adaptive response. All right, so dendritic cells, okay? So dendritic cells are sitting kind of at sites like barrier sites. So they're in your intestines, they're in your skin. And they'll phagocytose something from their environment. A lot of times, it's a foreign antigen if you're getting infected by something. So they grab it from the peripheral tissue and they hide it away inside. And then where do they go? Lymph nodes, good, regional lymph nodes. And they go via the lymphatic system, not by the bloodstream. That's important to know. So they go by the lymphatics, okay? So Cassie kind of talked about this already, but in lymph nodes, they present their antigens to naive T cells. What happens when you get a match with a T cell receptor and an antigen on MHC? You get proliferation, right? That T cell gets activated and stimulated to proliferate because we found a match, right? We got to go now. We got to make a lot of copies of this T cell to go fight this infection, specifically, not with the adaptive response. Innate isn't sufficient. So, and at this point, once they proliferate, they become those effector T cells that are going to go fight the infection. So, so where do they go? I mean, they go to the site of infection, right? And they go through the bloodstream. Now they don't go back through the lymphatics; they go through the blood. And a few of them will become memory T cells, just as a side note, but that's less important than the effectors for us for right now. So what happens now that we have our effector T cells? So let's say those effector T cells were mostly CD8. So what happens when those CD8 T cells recognize antigen bound on MHC1? What do? Kill it. Good. Yep. And perforin granzyme or fast ligand. Okay. This requires IL-2. Okay. From Th1 cells primarily. Okay, what about CD4 T cells? This is a little more tricky. So there's three different things that kind of happen in order for the T helper cells. Okay, the first thing is they differentiate, okay, depending on the cytokine environment. This isn't true for Tregs, right, that leave the thymus already differentiated. But here, those T0 cells, they get to the site of infection and then they figure out, okay, what do we need, depending on the cytokine environment. Okay, so Th1, Th2, Th17. Okay, and then next they will secrete cytokines, okay, to direct the immune response after they've differentiated into 1, 2, or 17. And then this is a thing with Th2 cells. Some of them will go back to lymph nodes and tell the B cells to do class switching or somatic hypermutation, okay? And we'll talk about that a little bit more, but know that it's mostly Th2 cells that are involved in B cell activation. Okay, this is just a picture from Dr. Drescher's lecture, just kind of summarizing everything that I talked about. So if you like pictures, this is a good one to kind of get yourself familiar with the process. Okay, so this is important. Okay, so upon activation by a dendritic cell, a naive T cell changes the receptor for which cytokine from low affinity to high affinity. What cytokine is now very high affinity for these effector T cells? Anybody? It's kind of a tough question. IL-2, good, great. Yeah, that makes sense, right? Because IL-2 is going to help them survive. Okay, and now that we've made our effector T cells, we found something that works. We don't want those cells to just die. That would suck. So, and yeah, why is this important? Yeah. Okay. This is another picture from Dr. Drescher's lecture of that receptor kind of becoming high affinity. Does that make sense? Okay, we're going to hammer IL-2 until the cows come home. Okay, it's important. Okay, let's look at cytotoxic T lymphocytes a little more. So this is the same thing, just IL-2 is especially important for those cytotoxic T cells to maintain their survival. Okay, So now we're going to talk about which cytokines induce differentiation of the different T cell subtypes. Okay, this is important. So which cytokines promote differentiation into Th1? There's two of them. Good, IL-12 and interferon gamma. And remember, IL-12 is produced by those dendritic cells and macrophages, and then interferon gamma mostly by the NK cells. Good. Okay, what about Th2 cells? What cytokines do differentiation to Th2? This is harder. IL-4, there's one more. 13, very good. IL-4 and 13. This is mostly done by mast cells, and this will make sense when we talk about Th2, what they do. They're mostly against like parasites and helminths, but now they just do allergies because those aren't a problem anymore. So, which kind of sucks. Okay, so now what about Th17? This is hard. If you guys don't know this, that's okay. They probably won't ask you, but there's 4 of them. 23, 6, yep. TGF beta. And the last one's really generic. IL-1. So these are details, but I would say Th17 is a little less important, but definitely know the Th1, Th2. Okay. And there's a picture for you if you like pictures. Okay. Okay, now we're going to talk about Th1 cells a little bit more in more detail. Okay, so I kind of mentioned this, but they're really good against intracellular pathogens, so bacteria and viruses mostly. So there's multiple different cytokines produced. What does interferon gamma do? What's its big job? Has to do with macrophages. Very good, yep. Specifically M1 macrophages, okay? It induces differentiation from, I think they're like monocytes at that point, and then they differentiate into M1 macrophages, which have increased killing capabilities. And these M1 macrophages also release more pro-inflammatory mediators. So TNF, IL-1, IL-12, and IL-12 will kick back and induce more Th1 differentiation, okay? What's the other cytokine produced by Th1 cells? We talked about it a lot. IL-2. Okay.
When in doubt, just put IL-2 as the answer.
Yeah, if you don't know, IL-2. Okay, so IL-2, as we know, T cell differentiation, particularly CD8 T cells, and survivability, viruses, right? Okay, so this is just looking at that M1 macrophage activation. This is just everything I said. The important thing to know is look at this interferon gamma here being released by the CD4 Th1 cell. Okay, and then now this macrophage is looking real mean, ready to do some damage, right? Okay. Okay. So next, Th2 cells. Okay, Th2 cells are really good against extracellular pathogens, but specifically parasites and helminths was kind of their original design. Okay, so IL-4 is a big one produced by Th2 cells. What does IL-4 do? It does 3 things. This is harder. Increases peristalsis. Yep. Yep. B cells. What class switch does it promote? This is a tough question. IgE. Very good. Yep. And then the last one is M2 macrophages. Okay, so it's the opposite of M1. So yeah, that class switch to IgE. And IgE was originally designed for the parasites and helminths. Okay, attached to mast cells. And then the increased GI motility and mucus secretion, that should make sense, right? Because we've got a big worm in our gut. We don't want, we want to protect our gut from that worm. So we're going to increase mucus. So there's a greater barrier between it. And then M2 macrophages are basically the opposite of M1s. They do important in anti-inflammatory responses, wound healing, fibrosis. It's important to know that during an infection, your Th1 response will be replaced by a Th2 response at the end. And that's mostly due to the macrophages. Okay, so initially you're trying to kill everything and then you've got to clean up the mess. Okay, and cleaning up the mess is done by the M2 macrophages. Okay, next is IL-13. It does 2 of the same things that IL-4 does. So mucus secretion and M2 macrophages. Okay, it doesn't do class switch. And lastly, IL-5 is a big one. So what does IL-5 do? Eosinophils, very good. Yep, it's also a chemotactic for eosinophils. And then what else does it do? It has to do with B cells. It's tough. Class switch to IgA. Okay, So class switch to IgA. So, and that should make sense because where's IgA found? It's at mucosal surfaces, right? So we got to buff up our IgA, which will help us, particularly in the gut, keep those pathogens out. Okay, neutralize them. It's a big, ugly picture, but this is all the different functions of Th2 cells, but it's basically everything I just said in picture form. Okay. Okay, this is important. This is looking at the M1 versus M2 subtype. So M1 macrophages, right? They're Th1 response, killing inflammation. M2 macrophages is cleanup, anti-inflammatory. Importantly, these M2 macrophages will release IL-10 and TGF-beta. IL-10 is also released by Tregs. Okay, they're basically anti-immune response, anti-immune system, and TGF-beta is also an anti-inflammatory. Okay. Okay, Th17 cells. Now this is tricky, okay? These ones are kind of the less well-known, but I think it's useful to know that they're really good against extracellular pathogens and mostly extracellular bacteria and also fungi, okay? We haven't really talked about that, but there's a few cytokines I think you should know that are produced. So the main one is IL-17, which should make sense, right? Th17, IL-17. So what does IL-17 do? Yep. And then there's actually three things. I keep doing that to you guys. I guess I did. It has to do with neutrophils. Okay, so it promotes neutrophil like longevity, and it increases the neutrophil response, which makes sense, right? Because neutrophils are really good against extracellular bacteria because they grab them, eat them, and then kill them. So, and then also there's antimicrobial peptides that are produced like defenses. Yeah. Oh, it was promoting inflammation. So I kind of combined them actually. So yeah, inflammation. And those are kind of the same thing because neutrophils are inflammation in some sense. But yeah, so. What about IL-22? What does this do? Very good. Endothelial barrier rigidity is what they call it. And then it also does antimicrobial peptides. Okay. That's not as important, but the epithelial barrier rigidity should make sense. You don't want any more foreign invaders getting inside of your body. What about IL-6? Generalized inflammation. Very good. Okay, so that's kind of what Th17 cells do. Neutrophil response is the big thing, the big buzzword with Th17s. Think neutrophils. Okay, this is just another picture. And then that other little factoid, the IL-23 thing, IL-23 is required for Th17 survival and maintenance. Okay, it's kind of like IL-2 in CD8 T cells. IL-23 is produced by M1 macrophages. I don't think you need to know that, but that's just an example of kind of the complex interplay between these different T helper subsets. And I will say, Th1 cells inhibit Th2 cells. Th2 cells inhibit Th1, and it's just by the cytokines they produce. So interferon gamma from Th1 will inhibit Th2. I think it's 3, 4, and 10 or 13 or something like that. Whatever Th2 cells produce. Yeah, 4, 13, and 5. Yeah, those inhibit Th1. So Th17 is more complicated. Okay. So summary. Okay. Some high yield factoids. So which Th subtype is involved with B cells? 1, 2, or 17? 2. Good. Yep. So Th2 cells can either do IgE class switch by IL-4, IgA class switch by IL-5. Okay, and they do this in the lymph node once they get there. Okay, and that's how. So there's that. It's kind of that. It's like a secondary signal that they send to the B cell while it's bound to the B cell. Okay. Note that they can also do IgG, but the mechanism by which they do that is a little more complicated. Okay. So which Th subtype induces CTL-mediated cytotoxicity? Which one? Good. Yep. By IL-2. IL-2 again, just keeps coming back. So which Th subtype does macrophage activation? Yeah, trick question, right? Both, okay. Well, that doesn't make sense. Both Th1 and Th2, not Th17. Okay, so what do Th1 cells, which type of macrophages do they do? M1, good. What do M1 macrophages do? Kill, good. Yep, and they release pro-inflammatory mediators. What about Th2 cells? What macrophages do they do? M2, Good. What do M2 macrophages do? Fix, clean up. Yep. Anti-inflammatory, tissue repair, fibrosis, phagocytosing, all that pus from neutrophils. Okay. Okay. A couple more high yield facts that might get you a point somewhere. Okay. These are kind of related, but also just kind of not. So which two antibody classes do opsonization? Okay. We didn't really talk about antibodies. Good. Yep. IgG, IgA are the main opsonins. Okay. What about this thing called antibody-dependent cellular cytotoxicity? There's two cells of the innate immune system that do this. Does anyone know what they are? NK. What's the other one? Mast cells. Okay. NK cells and mast cells do this ADCC. NK cells, well, we'll talk about it. Yeah. Okay. So they love this receptor. So what receptor on NK cells allows them to do this ADCC? Fc gamma R3. Yeah. Okay. That comes up a lot for some reason. So this binds to IgG, right? And IgG is already pre-bound. It's opsonized the bacteria, right? It's bound to the bacterial surface. And then the Fc region of the IgG will bind to the NK cell via this FcR3, and then the NK cell can release its perforin granzyme, right? Yeah. Okay. Oh, I wrote it right there. So how about mast cells? What receptor do they have? Fc epsilon R1. Good. And now this binds IgG or IgE. And this is important because IgE does not have to be pre-bound. This is why we're going to talk about hypersensitivity reactions. Type 1 hypersensitivity occurs very quickly because IgE is already pre-bound to your mast cells.
That's really high yield to know. And
yeah, we'll talk about that a little more. I'll give you some pictures. So that receptor is important. And then mast cells will release histamine and heparin. Okay, those are their two big things. And those just cause local inflammation, vasodilation, itching. So nasty stuff. Okay, so this is looking at the NK cell activation. So you can see IgG is pre-bound to this bacteria here. Here's that Fc gamma R3 comes along, binds, and then NK cells release their stuff, their juices onto the bacteria. So, oh yeah. Are you guys okay to keep going? I want to be respectful of your time. Okay. All right. There are some more memes. So it's not going to be all not fun like this. Okay. So mast cell activation. Now look at this instead. So imagine this as being pre-bound. Okay. And then you have an antigen comes along, cross links to IgEs. Okay. And then mast cells will degranulate histamine, heparin. This shows complement activation here, which can happen too. But histamine, heparin are the big things. Okay. Briefly about immunodeficiencies. Apologies. That's a rough one. But if you can make it through that, you can make it through med school. So congrats. So don't be doing that. Okay. Okay. What is immunodeficiency? So immunodeficiency is basically just absence or malfunctioning of any part of the immune system. Okay. So you can divide immunodeficiencies into primary and secondary. So here's a list of primary immunodeficiencies. I'm not going to talk about all of them. I'm talking about DeGeorge syndrome because it's really high yield. But know that primary immunodeficiencies are due to a genetic mutation present in the host. Okay. And then secondary immunodeficiencies are due to some external factor, whether it be HIV or malnutrition or even radiation therapy can damage your cells in that way. So there's a really good RX brick on immunodeficiencies if you're interested in reading up a little more, because I looked at the slides and it's kind of just a list. So if you can find external resources like this RX brick, it doesn't have everything that you put on there, but it has the high yield ones. Okay. And a lot of those immunodeficiencies aren't going to come up again, just being honest with you guys. DeGeorge syndrome will. That's why we're going to talk about it. But just so you know, and they're also really rare. Immunodeficiencies most of them are pretty rare. Okay, except like IgA deficiency, which is really common, which we're going to talk about right now. Actually, first, this is like a picture from first aid, being visual, having just an idea of where all these different immunodeficiencies are impacting, particularly the adaptive immune response in this case, is helpful for me. So IgA deficiency first. Okay, this one is the most common primary immunodeficiency. Just tuck that away somewhere. It's the most common primary immunodeficiency, IgA. It's really common, relatively. It's thought to be due to defective class switch of B cells to IgE or IgA. And remember, IL-5 is the cytokine that does that from Th2 cells. So that's kind of the thought, but people aren't really sure exactly how it happens. Lots of different mutations are probably contributing. Most patients are asymptomatic with this, but if you do have symptoms, it'll probably be along the lines of mucosal infections, which should make sense, right? Because IgA is sitting in your mucosa, protecting you from stuff. Pulmonary GI infections are two big ones. Treatment is antibiotics if you get infections. There really isn't too much beyond that. Okay, DeGeorge syndrome. Now this, I promise you this will come up again and again and again, and this picture will come up again and again, this one, so right here. I'm gonna introduce this to you guys. I don't think you have to know all this at this point, but it's really high yield for step, DeGeorge syndrome. So what is DeGeorge syndrome? It's a T cell immunodeficiency, okay? And the reason for this is you guys are gonna talk about like head and neck embryology. There's something called a pharyngeal pouch. And in this case, it's pharyngeal pouches 3 and 4 that normally descend and differentiate into the thymus and the parathyroid glands. But for some reason, in DeGeorge syndrome, this doesn't occur. So you end up with an absent thymus and absent parathyroid glands. Does anyone know what the parathyroid glands do? What do they do? Yeah, they secrete parathyroid hormone, which does calcium. It increases calcium levels. So these patients will often have hypocalcemia. That's just a little fun fact, but you don't have to know that right now. They love this mutation also. So chromosome 2211, okay? It's like this micro deletion of chromosome 2211. I swear it comes up all the time. So even that phrase, micro deletion, what? So, okay, so know that. So the symptoms, this is from first aid, catch 22. C is cardiac abnormalities, and cardio are going to talk about a lot of different congenital abnormalities. One of them is tetralogy of Fallot, which is a big one with DeGeorge syndrome. You can also see abnormal face structure. So the big thing is mandibular hypoplasia. So if you look at this, this kid's mandible, it's pretty small, right? He's got like a kind of a small retrograde mandible there. Thymic aplasia, this is the one we care about right now. This causes a T cell immunodeficiency, because you can imagine your T cells aren't maturing in the thymus if it's not there. So cleft palate, another big one, and that's the C. And then hypoparathyroidism, which we talked about, because your parathyroid glands are gone. So you get hypocalcemia. Parathyroid hormone acts on your bones to reabsorb calcium. You don't have to know that yet, I don't think, but later. This is just looking at where DeGeorge syndrome is a problem. So this is your immature T cells in the thymus. They can't become those naive T cells. Okay, that's it for immunodeficiencies. Take a look at that first aid brick if you want to learn a little more about that. Okay, so this is my meme for this. I don't know if you guys watch Breaking Bad, but I've always wondered that too, you know. Do they just like put like little pieces of nuts like in all their stuff? I don't know, but oh, so they're not dropping in little parts of peanuts into their food. Okay, I've always wondered that, but that makes more sense. I feel like that would be kind of weird. You like this idea better? Me too, especially if it was him. Might get a little something special too. Anyway, so. Okay, so hypersensitivity. Okay, this categorization will come up a lot. They like to say, they'll like give you a disorder and say, what type of hypersensitivity reaction is this? And so it's useful to know the different types. And we're going to talk about, there's a good RX brick on this, but we're going to talk about just the kind of overview of the different types. There's 4. So let's talk about type 1 hypersensitivity first. So 1 to 30 minutes after exposure. This occurs very quickly, and it's mediated by IgE, bound to mast cells. And if we remember, IgE is pre-bound to mast cells, so that's why it occurs so quickly. All you need is antigen. And all it needs to do is cross-link your 2 IgEs, and then the mast cells will freak out and shoot out their histamine. And histamine is what mediates the hypersensitivity. So what does histamine do? It does 3 things. First one is bronchospasm. Okay, so think about if somebody eats a peanut and then they can't breathe. So your bronchioles are constricting, right? That's a big one. The next one is vascular permeability. So it makes your vessels really permeable. So you get edema, like swelling, and itching also. So you get like hives. I don't know if you guys have had hives before. It's not great, but that's the histamine. And then lastly, they relax vascular smooth muscle. So you get severe hypotension in a systemic response. And this can cause shock. And what shock is you're just not getting enough blood to your tissues. That's just a generic term for shock. That's what that means. So anaphylaxis is a severe systemic mast cell response to an allergen. So it's like severe type 1 hypersensitivity. So what do we give people with anaphylaxis? EpiPen, right? Epinephrine. Epinephrine combats all three of these. It causes bronchodilation. It also causes hypertension. It constricts your vessels. And so it kind of helps restore fluid balance. So EpiPen is the treatment. Okay. So let me, okay, so this is important. So yeah, you need to be re-exposed to an antigen to develop type 1 hypersensitivity. The first time you get exposed, your B cells have to produce that IgE that's going to go find the mast cells, okay? So the first time you're exposed, the IgE gets produced, then goes to the mast cells, and then your mast cells are ready for next time, okay? And then the next time is when you get your type 1 hypersensitivity. Okay, any questions about type 1? It's pretty straightforward, I think. Everyone has allergies to something these days, it seems, including me. So, okay. Type 2 hypersensitivity, okay? So this is in a time frame of about 5 to 8 hours. And this is where I'm not clear because I'm not sure, usually it's an autoantibody. So I'm not sure how they're knowing like when you make the autoantibody. I'm not sure, but anyway, that time frame seems to be important. And what it does is it will bind to a self-antigen, usually IgM or IgG, and the antibody, and it causes one of three things to occur. So the first big thing is complement activation, okay? So the autoantibody, you guys learned about complement last week, right? So IgG and IgM can both activate complement. Next is opsonization. So this is important, like with your red blood cells. So IgG will stick to your red blood cells if you have an autoantibody to something on your red blood cell, and then your spleen will eat it. And then you'll lose red blood cells in your spleen by that way. And then lastly is some sort of abnormal physiological response. And so we'll look at kind of all those in more detail. So an example of complement-mediated damage is something called Goodpasture syndrome. Goodpasture syndrome affects your lungs and your kidneys. So the Goodpasture syndrome will cough up a bunch of blood and they'll pee out a bunch of blood because the autoantibodies are binding to the basement membrane in those organs. And it just causes a bunch of blood complement activation and then blood to leak out. And then you cough it up, you pee it out. So that's an example of complement. And then so opsonization and phagocytosis. So hemolytic anemia is one. So opsonization and it also does complement. So it lyses the red blood cells at the site by complement, or they'll go to the spleen and they'll get phagocytosed by the macrophages in the spleen and you'll lose blood cells that way too. And then lastly is like an abnormal response. So sometimes, if you guys have heard of myasthenia gravis, you get autoantibodies to your acetylcholine receptors in your muscle, and you can't get acetylcholine to bind because the receptor is being blocked. So you end up just not being able to contract your muscles because acetylcholine is that stimulating signal. So you just get paralysis basically. And then also there's thyroid stimulating hormone. You can get hyperthyroidism. Because sometimes the autoantibodies will activate the thyroid stimulating receptor and you'll get excess thyroid hormones. So it can go both ways. It can block it or it can activate, depending on the autoantibody. Any questions about type 2? Okay, so it's autoantibodies, complement, phagocytosing, or just causing some physiologic response. Okay, this definitely showed up on the quiz last year, so this is why I'm putting it in here. It's important to understand this process. So if you think about a fetus and a mom, so imagine a mom. You guys haven't done like hematology yet, but there's something called the Rh factor on red blood cells. So some people are Rh negative, some people are Rh positive. So imagine a mom who is Rh negative. So her body has not seen an Rh positive antigen before. And now she has a fetus who ends up being Rh positive. She inherited that or the fetus inherited that from the dad, right? And so during labor, oftentimes you can get, if it's like severe contractions, some fetal blood will end up getting into the maternal blood. And then the mom will say, okay, I've never seen this Rh negative, or I've never seen this Rh factor before. So I'm going to develop some autoantibodies to it. So then those antibodies will end up staying inside the mom. And if she gets pregnant again, if the fetus, those autoantibodies can attack the fetal red blood cells if they're Rh positive again. Okay, so it's the first pregnancy where the mom gets anti-Rh antibodies, and then the second pregnancy, if the fetus is Rh positive, those antibodies will go in and attack the fetus. So that's really bad. We don't want that. So what do? We end up giving what's called anti-D antibodies, and these neutralize the autoantibodies in the mom so that they don't attack the fetus. And this is done, I should know, this is done right after the first delivery. Okay, so it neutralizes them right away, and then the mom doesn't have to worry about it anymore. So that process showed up on the quiz last year. Okay, type 3 hypersensitivity next. Okay, so what are we looking at here? So another just time window, 4 to 6 hours. This is caused by what's called immune complex deposition. Okay, so it's similar to type 2, but slightly different. And the reason it's different is because of the location where the autoantibodies are binding. Okay, in type 2, it was at the site, it was at the tissue. Here, what happens is IgM or IgG bind antigens like in circulation. And then they end up just kind of complexing with more immune complexes, and they get big. And then these big complexes will end up just depositing in tissues. And once they're there, usually they activate complement. So they fix complement at tissues like type 2, but the source was these immune complexes binding in the blood and not the antibodies binding at the site where a complement is fixed. So it's a little different, but it's similar. And the high yield thing to know with type 3 is lupus. Okay, lupus is what you get is a bunch of autoantibodies. And a lot of those autoantibodies can be to circulating antigens. So you end up with a bunch of these immune complexes floating around, and they will deposit in tissues and cause a variety of issues, arthritis. There's the stereotypical butterfly rash. This picture shows up all the time, but that's immune complexes depositing in the skin, and then complement fixation, and then inflammation by C5a, C4a, C3a. This is a picture from first aid with those immune complexes depositing in tissues and causing complement activation. Any questions about type 3? So it's similar to type 2, but a little bit different. Okay, and lastly, we'll talk about type 4, and this will be my last little piece here, and then we'll be done, okay? So type 4 hypersensitivity, it takes the longest, okay? 1 to 3 days is a good estimate. And the reason it's called delayed type hypersensitivity for this reason, okay? It takes 1 to 3 days for the reaction to occur. And the reason for this is because it's mediated by T cells, usually CD4, but sometimes CD8. Okay, so CD4 response will be cytokine, because what do CD4 cells do, right? They release cytokines. So if a CD4 T cell encounters an autoantigen, they'll release a bunch of cytokines that'll cause local damage. CD8 T cells is a direct CD8 response to host antigens. This is much less common, but still can be seen. Okay, so when we think about different examples, so an example of a CD4 mediated response would be a granuloma in tuberculosis, and we're going to talk about that on the next slide. So it's a CD4 mediated type 4 hypersensitivity. And then examples of CD8 mediated would be like diabetes and then Crohn's disease. Crohn's disease is really nasty. We're learning about it in GI right now, but you get a bunch of damage in your GI tract, and it's mediated by CD8 T cells mostly.
It's also the type 4 hypersensitivity is also whenever you get like a TB spot test and they inject that stuff right under your skin and you can't get it read for 2 days, like 2 to 3 days. It's because it's a type 4 hypersensitivity reaction. So you have to let your immune system respond to it first, just if it helps you conceptualize it.
Yeah. And I'm going to talk about granulomas. This is high yield because granulomas, it's important to, if you understand how they form, it's not so bad. And it's really not so bad. Let me convince you here. So first thing that happens is we'll take the case of TB, okay? What is TB? It's an intracellular bacteria, okay? It lives inside macrophages. It can survive because it inhibits lysosome fusion to the phagosome. So TB will let itself get endocytosed, but then the lysosome components with all the toxic stuff, that doesn't get fused into the phagosome. So, and TB has special stuff it can do to stop that fusion. So it can just survive in these endosomes and it's its own little house basically inside the macrophage. Okay, so remember, intracellular bacteria predominantly promote a Th1 response, right? We talked about. Okay, so what happens next? So we have TB, we have a Th1 response, okay? Th1 cells will produce interferon gamma, right? Interferon gamma will act on monocytes, cause them to differentiate into M1 macrophages, okay? With me so far? So we have Th1 response, M1 macrophages with the TB inside them. Okay, next, these Th1 cells will also produce IL-2, okay, and that promotes the survival of themselves, basically. It's like a self-protective signal. And then this is a case where IL-2 is acting on a CD4 cell, not a CD8, but it affects both. Next, macrophages with the TB will produce IL-12, okay? That's one of the signals that they produce when they're converted to M1 macrophages. And then that IL-12 will act on more naive T cells to produce more Th1 subtype, okay? And then the cycle continues. Interferon gamma, macrophages, IL-12, Th1 cells, and it just maintains this sort of collection of macrophages with TB inside them, okay? And this makes a lot of sense when you think about it, because a granuloma is basically a wall, okay? It's a wall. It contains an infection inside it so that it can't leave. And so we keep kind of making more macrophages to contain that TB. And then these macrophages will kind of fuse together and form these giant cells, which is a pathologic finding on histology.
Yeah, and just to add to that, understanding that if you don't have IL-12 or interferon gamma, you can't form the granuloma or it will destroy the granuloma. So in order for the granuloma to be sustained, you have to have IL-12 and interferon gamma. I remember that being on the quiz or the final, asking like how granulomas are maintained.
Yep. So this is the way they're doing it, but I think it kind of makes sense. You know, it's a cycle, and it keeps the bacteria kind of contained. Okay, that's all we had. Unless you have any questions, if not, feel free to reach out. And best of luck to you.
Yeah, sorry it took so long, guys. I hope it was helpful.
Yeah, it went over time, but yeah, you'll do great.
Good luck, guys.
Good luck.
Also, on your way out, if you could sign our sign-in sheet, that would be wonderful. We kind of forgot to pass it around.